of the polymeric bioprobe. I will begin talking about our research background as the next slide. First, let me explain about Bioprobe. Bioprobe is a high-functional tool and used to explore biological function of biomolecules and cells. This is a model of Bioprobe. For example, after attachment of Bioprobe into some virus, antigen, antibody, and some other things, the labeled materials show fluorescence, which may be useful to determine the biological activity of the particular biomolecule or cell. A carbohydrate is an attractive biomolecule in order to explore biological activity, and the next slide introduces the function of the carbohydrate. The various carbohydrate structures are displayed on cell surfaces and play a role as a natural ligand between cells and their foreign substances. Schematic drawing is illustrating protein-carbohydrate interaction at the cell surface, mediating cell-cell binding, cell microbe adhesion, and cell antibody binding. In particular, the proteins termed lectins are the partners which bind to specific carbohydrate structures. The lectin usually has multi-binding sites on the surface, and cluster-type glycosides are used as an attractive biomolecule. The next slide will show you the glycocluster effect. It has been reported that glycolipids on cell surfaces can form glycolipid clusters named as RAPs, which are strongly recognized by saccharide binding proteins and complementary oligosaccharides. In general, interactions between saccharide and peptides or proteins are weak. Interactions are greatly enhanced due to the multivalent character displayed by the large number of repeating carbohydrate units along clustered carbohydrates. This effect is increase of activity is called glycocluster effect. This effect is used to drug delivery systems, biosensors, and tissue engineering. From the perspective described above, my research objective is development of synthetic methods for polymer bioprobe. Typical bioprobes have no molecular weight and act as monomeric bioprobes. Therefore, they have low sensitivity and one application per probe. But the polymer can show multivalent type probe and enhance the activity against foreign substances. In addition, we can choose monomers for the synthesis of bioprobe for many applications. We describe a conventional way for glycopolymers using commercially available reagents and for chloromethylstyrene as a starting material. And we also describe the biological variations of the glycopolymers having N-acetylglucosamine moieties for WGA as a model protein. This slide shows the synthetic plan of my research. Functionalized monomers are polymerized to give us the corresponding glycopolymers. From the next slide, I will tell you the synthetic pathways. We use 4-chloromethylstyrene as a starting material for an amide compound 2 converted into the amine derivative 3 by starting a reaction. Amine 3 was coupled with biotin in the presence of DMT-MM, which is known as coupling reagent to afford amidophore in 91% yield. As another functionalization, the synthesis of dancil monomer 5 was achieved in good yield by the condensation of amine 3 with dancil chloride in the presence of triethylamine. This slide shows the synthesis of carbohydrate monomer 11. Oxazoline was coupled with this diol in the presence of CSA to give glycoside 8, which was taken oxidation, followed by the condensation of amine 3 in the presence of DMT-MM. The coupling product was temporarily acetylated in order to purify to afford acetate 10 in 78% yield. Finally, quantitative deacetylation was performed for ester 10 to give us the corresponding carbohydrate monomer 11. Since the preparation of glycomonomer 11 was completed, we used the monomer for polymerization. Carbohydrate monomer 11 and an appropriate amount of acrylamide were polymerized to give us the corresponding glycopolymer 12A2D. The results of logical polymerization are summarized in Table 1. 12A is a mixture of compound 11 and acrylamide in the ratio of 1:0. 12B is 1:4. 12C 1:10. 12D 1:20. These are compositions. This is time. These are solvents. These are molecular weight of polymers. These are degrees of dispersion. And these are final yields of respective polymers. The couple of polymers were obtained and the biological evaluations of glycopolymers were performed. We selected WGA as a model protein, which is a well-known lectin and binds to glucanic residues. Thus, biological evaluation of glycopolymers 12A2D against WGA was preliminarily examined on the basis of fluorescence measurements. Association constant Ka was highest in the polymer 12D. The change by addition of glucanic residues was analyzed on the basis of Hill plot. Association constant Ka was estimated to these values by glucanic polymers. Interestingly, the difference in fluorescence intensity was the largest when polymer 12D was used as a substrate. This phenomenon indicates a significant change around the tryptophan residue located at or near the binding site of WGA. various densities of carbohydrate residues were efficiently synthesized from commercially available chloromethylstyrene. Biological evaluations of the glycopolymers for lectin were carried out, and the results suggested that these types of glycopolymers also display sugar clustering effect. धन्यवाद.
Any specific reason why you use the acrylamide?
アクリルアミドの他にってことですか？
何か理由があるかな？溶けやすいか
な？I use acrylamide as a model, so I don't think using other polymerized residue.
普通はアクリラマイドはトクシックじゃない。トクシックけども、アクリラマイドは。
アクリラミド以外にも使うかっていうことですか？今のところは考えてなくて、とりあえずモデルとしてアクリラミドを使っているので。
Any other? I think it is very clear presentation. So, how we can increase the efficiency of polymer by just coupling the monomer. Okay, I think we are running out of time, so yes. Thank you very much.